Hello and welcome to week 10. Week 10, we are going to be focusing on the cell cycle and mitosis. And you'll be able to understand what is going on in these beautiful pictures by the end of this chapter. We are going to be talking about the cell cycle and what are the various stages and what are the events that need to happen for a cell to be able to divide. Because of course we all start as one cell and divide and divide and divide, making exact copies of that cell as we grow into our multicellular organisms that we are. The cell cycle. So if you are a unicellular organism, one division is replication. You made a copy of yourself, you have an offspring. But if you are a multicellular organism like yourself, you still are going to need to make replicas of your cells. Again, from that development from a fertilized cell, from that just single cell into the multi-cell billions of cells that you are. For when you're growing, even after your arm is developed, you're still growing taller and longer. And then, of course, for repair, when you hurt yourself, when you get a cut. So this process of cell division is going to have two phases. Interphase that we see here, that's most of the time of the cell, and it's got three stages. There is G1. The G1 stands for gap. This is the first stage, the first gap. And this is actually where the cell does most of the cell things. When the liver cell is doing liver things, breaking down toxin, when your muscle cells are contracting and moving, when your cells are just doing their cell thing, that's G1. But then when they get the signal, hey, we need to grow or repair, then we're going to need to do what we just learned all about in last week's week 9. That's DNA synthesis. That's replication of the DNA to prepare to divide and be able to give one copy of the DNA to one cell and one copy to the other. So after the synthesis phase, there's the G2 phase. The G again stands for gap, and this is sort of preparatory. Now, the cell has made the commitment to undergo mitosis and to divide, so it's just preparing. So those are the three phases of interphase: gap 1, synthesis or S phase, and gap 2. And then the other component of the cell cycle is this mitotic or this M phase, which has got two parts, mitosis and cytokinesis. So by the end of this chapter, you should be able to understand what the DNA is looking like in each of these parts. G1, it's doing its cell thing. It's just the normal double helix. S phase, that's the DNA replicating, which we just learned all about to make those X-shaped chromosomes. And then that G2 preparing. And then we're going to learn about in prophase how that DNA is going to coil up. In metaphase, those chromosomes are going to line up, and then they're going to separate into ultimately two individual daughter cells in the process of cytokinesis. So this slide should look familiar. You can see the double helix. And in next week's topic, we're going to be talking about when proteins are going to again need to have access to that DNA and to those nucleotides. But for now, we're talking about how the DNA gets wrapped up super duper tightly into the chromatin fibers, and then that gets doubly looped up into these nice compact replicated chromosomes that we're going to see how they're going to get moved around the cell. Okay, so what we're talking about here in this cell division is we're talking about the cell division of somatic cells. So these are non-reproductive cells. So we're going to be talking all about mitosis and how the genetic material gets separated. We're also going to be talking about the separate process of cytokinesis. Now, this somehow is challenging to students because they happen at the same time. The very end of mitosis, cytokinesis is occurring. Now, gametes, this is the sperm and the egg, or the reproductive cells. We're going to learn how they divide in cell division through a process called meiosis. Now, this we're going to see is because it is important to have the amount of DNA when you are forming a zygote or making offspring when you are combining a sperm and an egg. So you take half the DNA from one parent and half the DNA from another parent to combine into one. So that's the process of meiosis we're going to learn about in a few weeks. Today, we're focusing on mitosis, which is happening in all of our non-reproductive cells, so all the cells other than your sperm and egg. We saw in DNA replication how one DNA strand gets copied into two. Now, those two copies are called sister chromatids. They are joined at the centromere, that's the location where they are connected with proteins that are doing that connections, including kinetochores, which we're going to see are going to bind to microtubules. But I want to just double check that everyone understands what is a sister chromatid. So you got one sister chromatid and another sister chromatid. These are exact copies of each other. They're exact replicas because in mitosis we're making an exact copy of a cell. So we are going to see how these two sister chromatids are going to get ripped apart into making individual new cells. So last week we learned how DNA got replicated to make one double strand become two double strands. And now we're going to see how those are going to get reseparated into two daughter cells. Now, this process is actually quite old, that we've seen it for a while. So even back in the late 1800s, people were staining chromosomes and watching how they were moving in cells and seeing how that DNA was separated. Of course, at this point, they didn't understand that it was DNA and that was the genetic material, as we talked about last week. So here, this video is a fantastic summary of everything you need to know. So you can either watch this video or you can watch the rest of me going through this. But for each of these stages I'm about to talk about, I want you to think about what's happening to the chromosomes. Where are they? How are they organized? What's happening to the microtubules? Are they getting put together? Are they moving? What are they moving? And then also the nuclear membrane. So the first stage is prophase. And again, this is the first stage of mitosis that happens after interphase, specifically after the G2 stage of interphase. So early prophase, what's happening? So the DNA has been nicely protected by that nuclear envelope, which is this one here. And so the nuclear envelope is going to begin to come apart. What's also happening is all that DNA which had been unwound is starting to get really tightly compact. It's got to be able to move. So just like when you move and you prepare to put all your belongings from one home to another, you put them in boxes to move them, right? To make it easier to move from one location to another. That's the same thing that the chromosomes are doing. They're condensing. They're making themselves be highly structured so that they can be easily movable. So the other thing that's happening in prophase is these centrioles, which are these organizational centers of these spindle microtubules. They were already duplicated in interphase, but they are going to start to move around, move from being next to each other to being on opposite sides. So what's going on as prophase continues? That nuclear envelope is coming apart, the chromosomes are condensing, those centrioles have moved to the opposite sides, and these spindles of microtubules, these are starting to form and reach out to the different chromosomes. Now, the spindles are going to reach out so that each chromosome is going to be attached by the spindle microtubules from both sides. So they're going to keep pulling each other and keep pulling each other. The microtubules are going to elongate and shorten depending on where they need to be moving the chromosomes to, until all the chromosomes line up at metaphase. So metaphase is the second stage of mitosis where all the chromosomes are lined up in the middle. The next stage, anaphase, those sister chromatids have been pulled apart. The sister chromatids have been pulled apart and are no longer together. Those microtubules are starting to shorten, and those chromosomes are getting pulled in to opposite poles of the cell. The next phase and last phase is telophase. Telophase is the exact opposite of what's happening in prophase. So now the nuclear envelope is starting to reform. Those chromosomes are starting to decondense, starting to open back up, and those microtubules that had been fighting with each other to line up those chromosomes in the middle, they are disassembling and starting to form the structures that they're always going to go to. So those are the stages of mitosis. I have some more of the information here about what I was saying, but we still aren't finished because we still haven't completed cytokinesis. So I've got more pictures. You need to be able to identify these cell cycle stages just by the picture. So looking at uncondensed DNA, knowing that's going to be interphase. Looking at prophase, starting to see that nuclear envelope fall apart, seeing those chromosomes condense. Some books talk about prometaphase in between, where again, those spindle fibers are moving those chromosomes, moving them around until they are lined up in metaphase. Then they get pulled apart in anaphase. And in telophase is the opposite of prophase. The chromosomes are opening up, the nuclear envelope is starting to form, and the microtubules, some of them are shortening and others are elongating for cytokinesis. So I do love this video. I'm going to pull it fast. You can see how those microtubules are pushing and pulling those chromosomes, lining them all up so that they can be sorted to make sure that they are going to get evenly pulled apart so that each daughter cell is going to be the exact same. You can watch it happen right here. They get lined up metaphase, and then anaphase is when they get pulled apart, and then telophase is when you're going to see that nuclear envelope starting to reform, and then cytokinesis is that separation. So I said cytokinesis, and that's separation. In animal cells, it's really quite clear where there's this cleavage furrow, which is actually this ring of microfilaments that kind of pinches it off, kind of like a drawstring on a bag. So that physically separates. So the cytokinesis is the separation of the cytoplasm and all the rest of the organelles in the cell. You can see that there's that contactile ring, and then there's also microtubules that are also pushing it apart. For plant cells need to have this cell plate. So that's that cell wall forming from vesicles that are bringing that cellulose and those different components of the cell wall to formulate that additional barrier between the two new cells. So you can watch this video. You can see some more pictures. You need to be able to identify the cell cycle based on photos. You need to be able to orient and describe what is happening to the chromosomes in mitosis, making these exact replica cells. So when we're talking about exact replica cells, that's important for us for when we get cut and when we're growing. But for bacteria are only one cell. So a bacteria making a copy of itself has reproduced, has made offspring. And how does it do that? We actually saw this last week as well. There is a single origin of replication for the single long circular chromosome that a bacteria will have. In that rolling circle mechanism, that DNA replication is going to go on both sides, copying that strand until there's two identical copies, and then it's going to split and pinch off. This is called binary fission. So that's the prokaryotic replication. So you can see there's only one chromosome. It's not undergoing mitosis. But you now need to understand these phases and be able to identify these stages of mitosis and the more and the longer phase of interphase. So you can give yourself a little quiz. Can you put these in order of mitosis? So hopefully you are able to see, you could have interphase where you see that nice nuclear envelope, you see that DNA that's fairly open, you see the nucleolus. So that's interphase. Then you see prophase, the nuclear envelope is coming apart. Those microtubules are forming. They're trying to reach out and join those chromosomes. The chromosomes are condensing. So this is prophase. Metaphase, those microtubules have pulled and pulled and pulled those chromosomes until they're lined up in the middle at metaphase, middle M metaphase. Anaphase, they get ripped apart. And in telophase, opposite of prophase, the nuclear envelope is forming. Those microtubules are pushing apart the cytoplasm, and the chromosomes are starting to open back up. And again, can you line them up? So can you identify them? As I just said, we got interphase, we got prophase, metaphase, anaphase, telophase, you need to know the stages and the phases of the cell cycle. What is happening to the cell? So how often this happens and how often a cell divide is going to depend on the type of cell it is. And theirs is all, of course, highly regulated. So deciding to replicate is going to involve lots of different controls called the cell cycle control system. And there's going to be controls that are coming from inside the cell and outside the cell. But either way, the cell needs to make it through various checkpoints. And what happens to a cell that's not following these checkpoints, that's ignoring those signals? That's cancer. So that's what all cancer is. Cancer is a cell that is replicating even though there are stop signs telling it not to. So there's 3 different major checkpoints, and these are all regulated based on the concentration of different proteins and different protein complexes with different cyclins and kinases. But I'm not going to expect you to know them at that level, but I am going to expect you to know that there are these various checkpoints and that cells need to be making sure that everything's okay before progressing. A cell doesn't want to progress if it's not going to be a healthy cell. So you first have the G1 checkpoint. Now the G1 checkpoint is actually in many ways the most important. This is where the cell is just doing its cell thing, being a skin cell, being an eye cell, a muscle cell, etc. And many cells, that's all that they need to be doing. When they're done, they can undergo a process called apoptosis and continue using their parts for other cells. So for many cells, this G1 checkpoint is the most important, and they won't continue on the cell cycle, that they won't need to undergo mitosis and replicate. So that's the G1 checkpoint. And there's also the G2 checkpoint, making sure that that DNA replication was fine. And then there's this mitosis, there's this M checkpoint that's during metaphase. Now, this checkpoint during metaphase is really important so that all those chromosomes lined up and are going to be able to be split so that each daughter cell will have one copy of each chromosome. So I already mentioned some things that are important for whether or not a cell is going to get the go-ahead to continue on and undergo mitosis. And those signals are going to come from inside the cell or outside the cell. And so those are things like growth factors, or if there's space for that cell to be continuing dividing. And when you get cancer, that's because they're not responding to this sort of regulation. So cancer cells have had some sort of mutation or a change in their DNA, which causes a transformation. So you have a cell and it gets a few mutations, enough that those signals that are normally giving that stop sign are being ignored. And there's lots of different ways this can happen. There's tumor suppressors, and those genes can get turned off, or there's things called oncogenes, and those can get turned on. Either way, in order for you to get a tumor, in order for you to get a cancer, you have to have multiple different hits to ignore those signaling molecules. That cell then will continue to divide and will start to take over and invade other tissue. Now, when cancer is considered to be malignant, that's when that cell goes from dividing in the place that it started, goes through the lymphatic or the blood system, so it can travel around your body, and then those cells will leave the blood or leave the lymphatics and go to another part of the body, so that you can have breast tissue that can go into your lungs or into your brain. So that's a malignant tumor. Now, one important thing to mention is that I said that at the beginning, in order to get a tumor, you have to have multiple different hits, multiple different mutations in the DNA of that cell. Why? Because you have your immune system that's normally circulating your body to make sure that cells that are acting weirdly or acting abnormally are taken care of and destroyed. We also have treatments, of course. Now, many of these treatments are not pleasant. So one of the treatments is high energy radiation that's going to damage the DNA, particularly in those dividing cells, so those cancer cells. There's also generic chemotherapies that target cells that are dividing. Well, and this is where you have impact on cells that aren't your cancer cells, but are just dividing, like you see for the cells making your hair, so people's hair falls out. Because this is a broad-based chemotherapy, so using chemicals to target cells that are dividing. But now there is good news that we do have some more personalized medicine to be able to look at what are the signals that are being ignored and seeing if we have drugs to specifically target those oncogenes or tumor suppressor genes that are being ignored. To end this chapter, I want to give a high recommendation to this book, The Immortal Life of Henrietta Lacks, which looks into this woman, Henrietta Lacks's life, who played a critical role in biology. She was the woman who died of cervical cancer, whose physical cells made the first cell line. So what is the first cell line? That means the cell that was able to continue growing in a culture dish. This was a tremendous, tremendous advancement in science and led the way for the polio vaccine and so much biological research since, all because of this horrific cancer that was able to grow and replicate so well that it could do it on a petri dish. So this book talks a lot about some of the really difficult and ethical issues that arose from essentially stealing this woman's cell without her knowledge. So I highly recommend this book. And with that, we are finished with week 10 material.